My name is Mike Robson. As Leon said, I'm chair of GCSE Biology here in the UK, where it's rather damp and grey autumn morning. I'm aware that you are from all over the world on these sort of occasions, and I'll do my best to sort of meet the needs of you for this particular course. The important thing to be aware of when it comes to the slides, let me just move to the next slide. Okay, is this is module 2 of this particular course. We had a very large number, nearly 100 people doing module 1 last week. If you were one of those people and you did module 1 last week and you've come back for module 2 this week, then there is a degree of overlap at the start and at the end because they are standalone modules. However, the stuff in the middle will be moving onwards and taking things in much greater depth than we did in module 1. There is a delegate booklet to download, but I am aware that some of you may well not be in a position where you can download or print things. And it is a little bit tricky because I'm asking you to look at a mark scheme and then look at sample answers and to see how many marks you would give it. And I can't share 2 slides on the screen at the same time. So I'll have to sort of show you the mark scheme, discuss it, and then we can look at the questions. I'll try and jump backwards and forwards where I can. However, if you haven't been able to download the delegate booklet, if you've got a bit of paper or a pen or something, you could scribble down the key points about the mark scheme so that when that disappears from the screen and you've just got the answers in front of you, then you won't be quite sort of disabled. So we'll do our best with that when we come to it. So the welcome and the introductions and a quick review of assessment objects will be very similar to Module 1. Then we'll go into much more depth on question styles and taxonomies and mark schemes and so on. And then at the end, the support from Pearson. I'll go into a little bit more detail with Exam Wizard, but otherwise that will be quite similar to what it was at the end of Module 1. So apologies if you were in Module 1 for a little bit of overlap at the start and end. But as I say, the stuff in the middle will be very different. So we're going to look at the assessment objectives for the qualification. We're going to look in detail at the various question types and to understand the mark schemes that go with them. And I think sharing mark schemes with students is something, that can be quite valuable. So your students also understand how it is that they can pick up additional marks. For instance, in an explain question, if they state the obvious, which is effectively a description, they'll probably get 1 mark, but they then have to go on because an explain question means, you've got to say, well, this happens because, and if you haven't written that part in your answer, and if you've looked at a mark scheme and you see how the marks are awarded, then you understand the difference between a describe question and an explain question. And of course, discuss and compare and things like that have their rules as well, where you can ensure you do pick up all the marks available. We'll look at practical questions as well. So we'll look at a different style of answers to various questions and what picked up the marks and what didn't. And then we'll go and have a look at the sort of support from Pearson again at the end. So just to make sure you're familiar with the chat room, could you just type into chat for me, first of all, whether or not you are new to this particular spec? In other words, you have not entered students for the exam before. If you entered students last June, then I don't know what the alternative to new is. I don't want you to put old. Yeah, just put in have taught it before or something like that. That's just let me see how many are completely new and haven't entered any students for the exam before. I'll just give you a second or so to find chat and to put that in. Okay, yeah. So it looks like the majority are new, but there's obviously a mixture. Already something's come up about modular. So let me just explain that. We don't touch on modular in this particular module. We did that in some detail in the first part of this course last week. So just to say the modular is an option for overseas students only. It's not an option here in the UK. The UK qualifications are strictly controlled by an organisation called Ofqual here in the UK. So the modular is not available in the UK, and the modular effectively splits the spec into two separate modules. The first part is topic 1 and most of topic 2. The second part is the last couple of units of module topic 2 and the rest of the topics. And then these are examined separately and in November and June, and these can be done in a modular way, i.e. you take them, you can resit them, and when you've done both and you're happy with the results, you cash them in and get the reward, rather than linear, where you get it, you teach the whole of the course and then get the 2 papers at the very end of the course. So I'll just mention that now, but we won't be covering it in any detail in the course. So just some details here about Pearson and a reminder about who we are. So we are the UK's largest awarding organisation and have been going on for quite some time, over 150 years, although I hasten to add, I personally haven't been involved quite that long. There's over 3.5 million students a year that are actually involved with the organisation. And just to say, in case you're unaware, the students will sit the exam and still, I don't know for how long, there's no immediate plans that I'm aware of, students will still sit down with a paper exam on a certain date and sit that exam, and then that paper will be sent off to the exam centre. These are then scanned, and the question papers are broken up into separate items, and then these are marked online by teams of examiners who have been standardized to make sure they're all marked to the same standard. And so in most cases, not in every case, depending on where it is in the world or what the particular qualification is, but in many cases, the whole of the paper is not marked with a red pen by one person. And then these marks are put together, and then the overall marks will be looked at in order to change those marks to a grade for that particular year. And that is an awarding decision that takes place by the principal examiners and the people that have been marking it. And if it turns out that one particular exam was a little bit harder than perhaps previous years, then the grades could be awarded at a lower mark. And if it turns out that the exam was easier than perhaps in some other years, then the grades might be awarded at a higher mark. So that awarding procedure takes place at the end. And that's happening with 3.15 million students a year in over 6,500 different schools or colleges. And it adds up to a staggering 10 million exam scripts that have to be dealt with over 70 different countries, with obviously different resources and different facilities and things in those different countries. Okay, so let's go on and look at the actual assessment objectives, which we did have a quick look at last week. So there are 3 assessment objectives, AO1, AO2, and AO3, and all the exam papers will reflect all 3 of these objectives. There's not a separate AO3 is practical, so there's not a separate practical exam or a practical paper. Each assessment will contain elements of those 2. I think it's quite important for teachers particularly to understand what these AOs are. Some teachers feel it's important the students understand them as well. And I agree with that to a certain extent, in that, they do have to be able to understand what it is they're doing well and what it is they're not doing quite so well. So if their sort of method of learning and of revision is to sort of, accumulate the facts, then they're probably going to do better on AO1. Their ability to apply those facts to situations they haven't seen before is AO2. So as you can see from this slide, the balance on the papers will be round about 40%, a slight tolerance, 2% each way, round about 40% on knowledge and understanding, AO1. You know, what is the name of this? Explain how this particular thing works. Equally though, 40% of the marks will be on AO2. And AO2 is about applying that knowledge and understanding, analyzing data, and evaluating stuff as presented. And this is really, really important. And this, I think, especially, you know, in the modern world, is where we're looking at things like transferable skills, where they've got the ability to look at data and say, well, is the conclusion drawn from that data? Is that a valid conclusion? Is it a reasonable conclusion? Is that conclusion backed up by the evidence that's in that data? And it's also important that students don't get thrown by AO2, that they don't suddenly think, you know, oh, there's a question on, I don't know, a zebra or a snake or something. And they go, well, my teacher never taught me about zebras or snakes. So, you know, this is going to be, I won't be able to do it. They've built up through the course an understanding that they will be presented with material, which they then have to analyze and interpret and look at and apply the knowledge that they've gained, not just recall the knowledge that they've gained. So 40% knowledge and understanding, AO1, recalling that information, 40% AO2, applying that information, and then the remaining 20% is the actual practical information. So that summarizes what I've just been saying, I think, about AO1. Okay, it's about understanding the content. It's about recalling the content. Some students feel confident with that. You know, they know they can label a diagram of the heart. They know the different parts of the digestive system or a flower or whatever. And so that if they're asked to name things on a diagram or to use those words in an answer, they feel fairly competent. They can do it. And you can help them to get to that position by obviously repeating. It's often a good idea at the start of every lesson to sort of repeat and test what you did last lesson. So you don't just start saying, today we're going to do this. You say, last lesson, we did this. What was such and such? Can anyone remember the name of so-and-so? What did it mean when we said this? When we were talking about diffusion, what was the direction of flow relative to, you know, right, okay, that was last lesson. What we're moving on to today is this. And so that little bit of repetition at the start of one lesson, it's almost like when you watch a series on the TV, they invariably sort of use up the first couple of minutes recalling the last episode before you then launch straight into this current episode. So testing with things like mind maps and flashcards, use the students to test each other. Say to, you know, just say to students, right, okay, if you're sitting next to somebody, who's student A, who's student B? Right, student A explain to student B about why enzymes are denatured at higher temperatures and what actually happens, and use the word active site in your answer. And so, you know, it puts the students on the spot, and then student B can be equally embarrassed by having to explain to student A what happened. And the kids get used to this after a while, and that embarrassment dies, and they understand that it's a good method of helping them to learn. So here's a typical AO1 question where it's named the structures labeled A, B, C, D on the diagram of the eye. Okay, clearly they either know that or they don't know that. They've learned it or they haven't learned it. There's not a lot of room for guessing. Some might be labeled on there for you. So you've got some sort of labels which are actual words. Perhaps they will come later on in the question or are referred to later on in the question. Here's another example of an AO1, this time a written one, an explain one. But, you know, to explain the changes that occur in the structures of the eye, that's where perhaps the Fourier answer on that diagram earlier would help you a little bit. That allow the light of distant objects. So we've got to pick up that it's not near objects, it's distant objects to focus on the fovea. So that's the structure that you can access there. And that is an AO1 question because it's based on knowledge. They're not applying it to a situation they haven't seen before. They are simply, they've learned how the eye focuses light, and so they're being asked to repeat that information. AO2, as you can see, is emboldened there in the first bullet point. AO2 is about the application of that knowledge. So maths questions will fall into this, where they have to use calculations or graphs, particularly analyzing data, tables of data, and then being asked to sort of interpret what's going on, describe what's been going on, but also then explain why they think this particular data is valid or not valid, evaluating it, discussing it, and as I say, it's important that the students, when they see something that they're unfamiliar with, don't think that they can't do it or don't think they haven't been taught properly. They're expecting this type of question to come with what they're doing. So those are AO2 questions. And here's a typical AO2 question. And just to illustrate what I was saying earlier about the describe or explain, if I'm not, this question is a maths question where you're asked to calculate things and then suggest an answer at the end. But if I was asking to describe this, then for instance, if I was asked to describe germinating seeds at 12 degrees Celsius, then I would say it goes up. And I'd probably get 1 mark in the describe question to say it goes up. I'd probably get more marks if I said it started at 0 and went up to 20 minutes. So I'm using the right units. Or it started at 0 and went up to 0.8 oxygen absorbed, brackets in centimeters cubed. In other words, I'm adding more data from the graph and makes that a better answer. And that would be a describe answer. If I'm asked to explain it, often the exam question won't say explain why it goes up, because that could be 1 mark. It might say explain why the time the oxygen absorption changes in germinating seeds as the temperature rises. So they'll use the word change. Okay, so you'll get 1 mark for saying, well, that changes, it goes up, it rises, as time goes by. So they get then asked to sort of explain what that's going on, or to compare it to the 12, compare the different temperatures that are shown in the graphs. So an explain might only be 1 mark for saying what's going on there, but then, sorry, an explain might be just 1 mark for saying what's going on. The rest of the marks will be for why it's happening, because it happens because of this. Okay, so AO2 questions, as I say, don't be put off by the fact that you haven't seen it. Just as a little aside, I was marking some papers, it wasn't this year, that might have been last year, about germinating seeds. And a lot of the weaker students started talking about bacteria. And I couldn't understand where bacteria had come from, because it wasn't in the question at all. And then I suddenly realized that they didn't know what the word germinating means. And because it started with the word germ, they guessed germs, bacteria. And that's what they were doing. And so it's often important to reinforce that sort of, terminology, diffusion, words like that. Make sure they've got those in their vocabulary, and they're not going to be thrown by words they're not familiar with in an exam question. So AO3 then, a more simpler one to understand. AO3 is quite clearly just on practical. So it could be the core practicals. And you're aware that the core practicals are detailed very clearly in the specification. And the assumption is that the kids have done those core practicals. So you could argue in a way, that's as it says there in the second bullet point, that you could argue in a way that some questions on the core practicals are actually AO1. because they're recalling when they actually did that particular question. They're not applying it. They're not using the practical skills. They're just remembering how they did a particular practical. And that's a fair argument, although I think we would probably keep it simple and simply say if it's on a practical topic, then it's AO3. And then you could well be asked as well, not just to recall the core practicals, but perhaps apply the knowledge that you gained in a core practical to a slightly different situation. So you might have tested food for glucose and then be given a context where you're asked about testing blood for glucose. Not what you did. You didn't handle blood in the labs and test it for glucose, but you have to appreciate that the test is the same and therefore describe how you would do it. And so in that context, you could argue there's an element of AO2 in there because they're applying their knowledge to something different. But nevertheless, if it's based on the practical parts of the spec, we would probably call it AO3. And so these can be, higher demand, especially when they're asked to sort of decide whether the results in a particular experiment are reliable and the conclusions that are drawn from those results are valid. And you can also, of course, be asked to design experiments. What would you do, design an experiment in order for you to do such and such? So the level of demand can vary with bits of AO1, bits of AO2, and bits of familiar core practicals and unfamiliar different practicals. So if I sort of play a student with this question for a minute, first of all, you can see it's part B. So part A of the question would have probably led me into the context of photosynthesis and to understand what was going on in some way. I wouldn't just be suddenly starting with part B of the question cold. So a student uses this apparatus to investigate the need for carbon dioxide in photosynthesis. Right, okay. So let me look at this experiment and get it into my head. I haven't rushed to read the questions. I'm getting the information into my head. So clearly there's a plant there where one leaf has been isolated in a conical flask, and they've put this chemical sodium hydroxide in. Now, do I know what sodium hydroxide does? Could I guess what it might do if sodium hydroxide is NaOH and carbon dioxide is CO2? Might that provide more carbon dioxide? Well, that seems unlikely if it's sodium hydroxide. Might it absorb the carbon dioxide? Well, that seems more likely, yeah. Okay, so I'm getting the information in my head. Right, what's the question? First of all, the command word is explain. So I need to get a because or something into my answer. Explain how the student could use this apparatus to show that carbon dioxide is needed for photosynthesis. Okay, so I think the because here would be is that because the air inside the flask has different carbon dioxide compared to the other leaves of the plant, and therefore if I pass those, test those two different leaves, then I might get two different results. And so the because bit is that I'm creating a different environment in that conical flask. And if I can explain exactly what the sodium hydroxide does, so much the better. So the student is told that after keeping the leaf in the flask for a day, he should cut the leaf into small pieces for testing. Suggest why this is a good idea. Well, here again, you're applying the information. If you cut the leaf up, you're obviously going to have several samples, so you can perhaps repeat it more than once, which would make the results, more reliable and your conclusions more valid. So all of that information under suggest, not necessarily, a right answer or a wrong answer, because the command word is suggest. So there could be a variety of different things that I could come up with on the marks. I also note, that the explain bit was 4 marks and the suggest bit is 2 marks. So I'd look at my answer and think, well, obviously the answer lines are not shown here on the screen, but I can get an indication. I don't have to fill up the space, but I'm getting is putting an indication of what I'm needed. So just one of the points that I think Roma's picked up there on and explain, you have to give the reasons for your description. Exactly right, yes. And as I say, that from a student point of view, just say to them, have you put the word because in there? Have you said, this happens, probably one of those 4 marks, this happens because the sodium hydroxide changed the nature of the air inside the flask. So the circumstances for photosynthesis for that leaf inside the flask is different to the circumstances for photosynthesis outside that leaf. And they get all full marks. I would know exactly what the impact and what that sodium hydroxide was doing. So you could argue in this question, although it's an AO3 question, you'd probably classify the first part, dotty 1, as AO2, because it really is applying to this situation your knowledge of what the role of carbon dioxide is in photosynthesis. So if it was just a photosynthesis equation, that would be recall. But you're applying your knowledge of photosynthesis to this situation as to what's going on inside it. So that's AO2. Okay. It probably the second question, the students told that keeping the leaf, suggest what that is very practical, isn't it? Why would you chop the leaves up? That's definitely, without any doubt, an AO3 question. And so you may get an AO3 question like this one as well, where you simply have to design an investigation. So again, you're looking at the original context. Breakdown of the dead plant material by microorganisms is affected by many factors, right? That sets a context. And again, notice this is question B. So there's been an A coming before it, which would be probably something about microorganisms or something that would have got me thinking along the right direction. Design an investigation. So clearly an AO3 design question to determine the temperature at which plant material is broken down most effectively. And a bit of giving you a clue, because it's a 6-mark question. Include experimental details in your answer and write in full sentences. Don't always have to write in full sentences. You can do it by bullet points. But because it's a practical, I'll show you something about bullet points in a few slides' time, which is worth thinking about. So clearly, I'm going to have to think about, well, how many different temperatures am I going to create? In what sort of circumstances are those different temperatures going to be? And then I would have to think, right, okay, well, if I'm varying the temperature, what are the things that I'd have to keep the same and to make sure that my results are suitable? So that's a typical AO3 question. Okay, so let's go over to you now and see what you think. So let's just do this in the chat room. So here's a question. Can you tell me whether you think that is AO1, AO2, or AO3? Notice again, it's part D. All these questions are just ones I've pulled out from exam papers. So, you know, I haven't shown you the whole question. So the stomach is an organ found in the alimentary canal. Describe the role of the stomach. So I'll give you just a minute to think about that and see what everybody's doing. Yeah, and I think we're getting very consistent results coming in there from everybody. Everybody's thinking it's AO1. And yes, you're absolutely correct. Totally agree with you. Clearly, describing the role of the stomach is something you have learned as facts, you have revised as facts, what goes on in there with the acid and so on. You've learned those facts, and then this question is asking you to give them back. So it's an AO1 question. Thank you. What about this one? Let's look at C.1 first. The scientist concluded that pollution reduces the growth of the shoots. Name the type of cell division affected by pollution in this investigation. So you're naming the type of cell division. So .1, what do you think that is? Okay, so clearly the question is on cell division. No, a couple of people said AO1, sorry, AO2, because it's sort of, linked in the first part to pollution and the growth of shoots. But nevertheless, no, I think really this is named the type of cell division. So at a certain extent, you've got a 50-50, haven't you? You could either go for mitosis or you could go for meiosis. At GCSE level, I probably don't know any other type of cell division. So if I'm naming a type of cell division and I don't know the answer, I've got a 50-50 guess if I have to guess. A point here about spelling. This is one of the classics. We tend not to mark things wrong. If the word of photosynthesis is spelled, Fotosin or something, it's fairly clear that they mean photosynthesis. It can't be anything else. So the student would still get the mark, even though it was misspelled. However, if you start writing sort of MEIOTIS, then it's almost impossible to tell, do you mean meiosis or mitosis? So we have to advise our examiners to come up with a rule and say, yes, you can mark things if they're phonetically, if what the kid has written is what it sounds like, photo, sin, the, cis, it couldn't be anything else. So in this particular case, the advice we give them is that if it's got a T in it, it means mitosis. If it hasn't got a T in it, they mean meiosis. So obviously, it's always better to make sure you can spell it correctly, but we don't mark it wrong just because it's not spelled correctly, as long as we can understand fairly what's going on. So yes, I agree. This first part would be AO1. And then to make sure the conclusion is valid. So the conclusion from the experiment, validity, the scientists control variables, the abiotic variables, when the seeds are germinating. Discuss two abiotic variables that needed controlled. So is that AO1, AO2, or AO3? If you can put in the chat box what you think. So you're asked to discuss as a command word, discuss two abiotic variables. So getting a bit more mixed response there this time, which is understandable, I think. There's no point giving you just easy stuff, is it? Yes, you're applying your knowledge. It's definitely not AO1. So yes, you're applying your knowledge. So it could be AO2. But in this context, you are applying your knowledge to a practical situation. So this question was classified as AO3 because it's in a practical context. Yes, I can see why you think AO2 because you're applying knowledge. But if you're applying knowledge in a practical context, then it's classified as AO3. And then the last bit as well, state one biotic factor that the scientist should control. So it's a biotic factor. I have to understand what a biotic factor means. I have to understand control, but it is in this particular context of this particular experiment. So that is AO3 also. So I think we'll do just a couple more of these. What about this one? Okay, so again, looking at this one, and we've deliberately picked some sort of slightly borderline ones. I can understand, and you may want to think it's a valuable exercise for perhaps for us as teachers to understand what's going on in the question. Might it end up confusing students if you ask them to do this exercise, because it is quite tricky to work out what's going on. Overall, well, I know this question was classified as AO1, but there's a very strong argument to say that it could be AO2 because you're saying this is a fungus based on corn. So effectively it's AO1 because you're just explaining how fungus feeds. So, that's knowledge about what fungi do, fungi in general. So it's AO1. However, is the application to corn, would the answer be any different if the word wasn't corn there? Probably not. So I think I'd probably go for AO1 on this one, but I do accept that there's an element of AO2 because it is applied specifically to corn. But overall, I think it's about how do fungi in general feed. That would get your answer. Okay, what about this one? 40% of the energy in the product and the producers, again, this is dotty two. I'll stop saying this. You understand there's always stuff before these questions. 40% of the energy in the producers transferred to decomposers. Only 20% of energy in the primary consumers transfers to decomposers. So 40% of the energy in the producers transfers to decomposers. Only 20% of the energy in the primary consumers transfers to decomposers. Explain this difference. Yeah, I think everybody's been very consistent on this one. We've had a few variables on the more confusing ones, haven't we? Which is understandable. But yeah, everybody's plotting for AO2 here. And I totally agree with you. It's not asking a specific recall of knowledge because it's not AO1. It's not in a practical context. So it's not AO3. And, obviously, as I said, that probably before this question, there would have been an actual flow chart or a food web or something to look at, or a pyramid of numbers or pyramid of biomass. And they're then asked to sort of explain what's going on from that. So yes, AO2. So using these different assessment objectives, remember we said AO1 will be 40% of the exam, AO2 will be 40% of the exam, and AO3 will be 20% of the exam. So using that is a valuable tool for the students to be aware of. So how can you help them? And perhaps more importantly, are they dropping marks on AO1 questions because their recall isn't good enough? Are they dropping marks on AO2 because they get confused or phased out by applying their knowledge? They think, oh, I don't know this. This is unfamiliar to me. Or is it that they just haven't picked up the basic sort of practical skills about what controlling variables is all about, and how to sort of set up a control experiment and how to use basic apparatus and tests that have come through. So we'll talk about Exam Wizard in a minute. So you can, select questions based on AOs or topics based on a particular core practical. You can pick exam questions out from the vast bank of past questions that there are now on that thing to do that. So helping students to self-identify the areas for development by filling in grids. And I'll show you one of those in a minute. Obviously, Results Plus, that tends to be after you've done one cohort of students and you're perhaps preparing for the next, to sort of say, well, that class that I taught last year, how did they do? What was their strengths? What were their weaknesses? How might I change things for the new classes coming through? You could access Results Plus there. And then the access to scripts is literally straight after results day. You can go straight in. And if you are surprised that a certain student perhaps got a lower grade than you were hoping for, you can go in, you can see how close they are to the boundary, and then decide whether or not it's worth thinking about a remark or something. So there's various reasons why you could use Exam Wizard. And if you haven't used it before, this is the sort of screen that comes up when you first go in. And you can particularly see the headings across the top there. So you can use it as a database of past papers. You don't have to keep a filing cabinet full or on your own personal PC, keep a copy of every sort of paper that's happened. You can access every past paper. If you wanted to find the June 21 paper 1, then you can just put that in the search and that's what will come up. You can build a paper yourself. So if you wanted to sort of build just a practical set of questions, or to build just AO1 questions or AO2 questions, or just all the questions you can find on the eye, because you've just taught the eye, or the heart, or you know, the carbon cycle, or whatever it is that you want to build, you can go by topic and build a paper. And then when you've built that paper, you can save it and put it into My Papers. Or if you're doing a more long-term job, you know, you can start, if you want to like build up your own mock exam and you've done like the first 3 questions, but you haven't finished it yet, you just store it in My Papers and go from there. So it's free, it's available to everybody, it's online, and you've got everything in it that you might need. And you can see there, when you build a paper, then obviously you select the qualification. You can select all units or just one unit. You can select an AO, or you can have all AOs. You obviously have to say what specification is you're looking at. You can pick on particular topics. You can pick a particular year. You don't have to select 1 or more years, but you could perhaps pick just the last 2 or 3 years of exams. You've got the freedom to do that. You can pick by a certain skill, like I said, so you could be looking at controls in practical exams or so forth. You can pick out just, you know, a particular series, so you can pick out the 2023 exams or the 24 exams, and then you can go for a question type. So you could make up a paper of all multiple choice questions, or all described questions, or all compare questions. So you've got a huge amount of flexibility with every past paper sitting there for you to go in, to have a play. I'd advise you do that sooner rather than later if you're not familiar with Exam Wizard. Just go in and have a play and then think about how you can use it as a way of accessing materials to help your students. The examiner's reports are also in there and the mark schemes are in there. So if you're building your own paper, don't forget to build the mark scheme at the same time you're doing it, or you might forget where you took various things from, and it makes it that bit harder to sort of get the mark scheme. And then, of course, you could sort of say, well, I think I'll use this question. You could then, you look at the examiner's report and say, oh, well, the examiner said that this particular question was very well done. So I would expect my students to do well on this one. Or the examiner might have said, very few people got full marks on this question. So you can say, right, okay, so what do I need to do with my students to try and make sure they would get full marks? So it does give you that flexibility. And then if you want to, you could give the students a sort of like a past paper and then give them something like this afterwards, a check grid, where instead of just saying, well done, you got 65% on the whole paper, thank you very much, put it in the bin now, and we'll move on. You actually drill down into that paper and say, right, so the first question in question 1 was worth 3 marks. How many did you get? Did you get 0, 1, 2, or 3? And that particular question happened to be an AO1 question. And then you can go through the rest of question 1. And then when you get to the end of question 1, You can look at the 19 marks that were available. The student can write in how many they got. So did they do well on this particular topic? 2.5 on the spec. You could look that up and remind yourself of what it was that they were doing. So this would have been, flowering plants and the role of xylem and water being absorbed and so on that. And there's practical in there as well. So you can look at the score, you can get an idea how well they did on that particular topic. They can get an individual score, and then you can drill it down further and say, well, did you get 5 out of 5 for the AO1 questions, but only got, 3 out of 9 for the AO3 question? So that's where your weakness was. That's where you didn't do so well in question 1. So you can do a grid like that to help sort of work through and think about it. So those are the sort of overall skills that you can do. Let's just look at the AOs specifically. So AO1. Again, remember we said this is factual knowledge. So simply making revision notes, mind maps, lists. I always found lists were particularly important. If I've got to label a diagram of the eye, let me list the labels. Even if I haven't got the diagram in front of me and I know where to put them, can I just remember the words? Iris, cornea, retina, lens, all those sort of words, just make a list of them and refresh my memory, then go back and look in the textbook, look at the diagram online. What did I miss? Did I forget the ciliary muscle? Did I forget something or other that was in there? Blank page revision. This is a technique where you literally start with a pen and paper and you say to the kids, right, here's a blank bit of paper, write down everything you know about photosynthesis. Okay, off you go, 5 minutes. And then they can sit down and say, right, sit down with the person next to you, cross out all the things that you both wrote down. Okay, now look at the things that only one of you wrote that the other one didn't. Were they right? Were they wrong? But why did the other person not think of them if they were right? Why did the other person think of them if they were wrong? So you can use the students to talk to each other to look at what they're doing there. And of course, when they're doing that, there's an element of teaching each other going on. And you may be the same as me, I don't know. But I certainly found that when I had to teach a topic, that's when I really began to understand it. When I had to explain it to other people is when I really began to understand it. And so give the students that opportunity to teach each other and to talk to each other about what they're doing. If you haven't done it before, they'll be quite embarrassed and find it quite strange to start with, but they very quickly get used to it. And it really isn't a sort of a problem for them after a while. Vocabulary, keywords. Okay, again, just keeping perhaps a vocabulary list would be quite important, especially with the less confident students. If they're scared of using the right sort of vocabulary because they feel they can't spell it or they're not sure exactly what it means. But, if you think of something like diffusion, osmosis, and you start talking about concentration gradients and osmotic potential, there's a lot of vocabulary in there which potentially might confuse the fact that they understand simply that if something's a bit more concentrated in one place and a bit less, if you put a drop of water onto your shirt, then the dampness will spread out from where there's a lot of water out into where there's less water. It doesn't go the other way around. Your wet shirt doesn't all concentrate into one little drop of water and the rest of the shirt becomes dry. So understanding at various levels can be quite important for them on key topics like photosynthesis, digestion, as well as the processes like diffusion and so on. So vocabulary lists can help. So that's all potential ways of reinforcing AO1. AO2 really is a question of practice. So, any sort of data analysis. It can be from the newspaper that morning. It can be from the television the night before. It can be from stuff going on in school about school dinners or anything. Anything that is data as presented, and then ask them to analyze that data and to think of questions about that data. And as I said earlier, these are transferable skills. These are valuable skills, not just for their biology exam, but for their other exams as well, and for life in general, to be able to be quite critical about the data that's presented to you. How big was the sample? You know, when they say 100% of people did such and such, well, if there's only one of them, that was 100%. If there's only five of them, how valid is that conclusion if all five of them did the same thing? Where did they come from? How old were they? Were they male or female? You know, what was their background? Those sort of questions are transferable skills when it comes to analysing data. And that can be built in, not when they start their GCSE courses, but that can be built in, as it's saying there on bullet point 3. That's something that can be built in the whole of their education. Graph skills, numerical skills, analytical skills, you know, these are transferable skills that can be built in. And it builds the confidence in the students to take something that's unfamiliar and to work with it. So evaluating, seeing both sides of the argument as well, not just necessarily believing. You know, it wouldn't be wise for me to start mentioning any of the politics around the world that goes on, but you hear a whole lot of stuff being presented as fact, and then you have to question, is it really fact? And these transferable skills can be built by this sort of analysis. If they're finding things like, describing things difficult, then you can build up initially by giving them lots and lots of scaffolding. So rather than just saying, here's a blank bit of paper, describe what's going on, you can say to them, right, okay, if you're presenting with this graph, well, what's on the x-axis? What's on the y-axis? What's the overall trend shown on the graph? Is there any peaks or troughs? What are the actual units that are used? What conclusions could we draw from the first part of the graph, then the second part of the graph, then the third part of the graph? What key words should we get in when we're describing what's going on? So that's scaffolding. You don't just say, sort it out, here's a blank bit of paper. You give them structure to start with to try and work out what they're going through. Okay? So data exercises like that, the kids enjoy them. They can be just something you start the lesson with. You know, did anybody, remember last week we did so-and-so, so-and-so, or remember a few weeks ago we did so-and-so, so-and-so? Did anybody see on the television last night that they were reporting back about the percentage of people that are using vapes? You know, here's some data, about analyzing whether nicotine is addictive or not, and how many vapes have nicotine in them and how many don't. And are the colors targeting different age groups? You can look at data. It's not to do with the spec, but analyzing that data. And then you could say, well, what do you think the effect of those vapes are having on your lungs? What do we know about the structure of the lungs that might be affected by the gases that you're sucking down into them? So that's part of that AO2 and the transferable skills. So using those sort of, different types of analysis, any sort of data will do. This is one that we just sort of found, very obscure, about different types of shrews. If you're not familiar with shrews in your country, they're sort of like a small mouse. So it's looking at the body mass and relating it to oxygen use. So here's some scaffolding. I won't go through them all, but here's 6 different questions based just on that one table of data. Okay, so you could just ask them, here's the data, discuss what you think the relationship is between body mass and oxygen use. Or you could structure it by saying, describe this, then describe the next, then look for the pattern, look for the relationship. So, hopefully the first suggesting on that screen, where you're just asking them to discuss, would be when the skills are well developed, or the alternative, where I've given you sort of 6 different steps there, is where you put in scaffolding to allow them to take it through step by step. And so here, when we get to AO3, clearly, what makes science lessons different from geography or history lessons is the fact that science is a practical subject. There are core practicals which you're expected to do hands-on, handling the apparatus, using the microscope, adding things to agar plates, examining seeds and flowering plants, and perhaps even doing simple genetic experiments with flowering plants, extracting DNA. These sort of practicals, which are very doable, and there's no doubt about it, if the kids have had hands-on experience of doing these things, then they can cope better than if they haven't, or if they've just watched a video or whatever. Learning the vocabulary, accurate, variable, repeatability. Could I repeat this experiment from the data that you've given me? And, often the kids might say, oh, my experiment didn't work. Brilliant. No such thing as an experiment not working. You got what you got. The question is, why did you get what you get? Why is it not what you're expecting? Was it too concentrated? Was it too hot? Did you forget to add a certain ingredient? To a certain extent, it's like a recipe. It's like a cookery lesson. They're putting stuff together and looking for an outcome at the end. And it's a question of, do they need a recipe to do it? Do they have to follow it step by step exactly? Or can they interpret and use and adapt and design to come up with? So it's like, what's the difference between an experienced chef and somebody just following a recipe to cook a meal? So that sort of understanding of practical skills is important. And then being able to use the maths and things like that to interpret the stuff afterwards is equally important. Again, when it comes to planning practicals, to start with, if you just say to them, right, okay, we want to test to see if carbon, like that earlier thing, we want to see if the amount of carbon dioxide in the air affects the rate of photosynthesis. Design an experiment. That's a very demanding thing to throw at the kids if they haven't done it before. But you can give them this level of scaffolding. So you can say, right, we're going to do this practical. Okay, what are the important words? What is the independent variable in this? What variables do I need to control? And you can see when you mark questions on these, what the kids have sort of understood. They're straight away into sort of things like, well, if it's in a room, what was the temperature of the room? What was the light level in the room? Would that have made any difference? If one student was doing the experiment next to a radiator and the other one wasn't, if one student was doing it next to the window and the other one wasn't, if it was left overnight, or if we came back next lesson to test our results, what difference would that make? These are the sort of questions which the student should be asking, and you can prompt those questions by giving them something like this, filling in the blanks in sentences. So you're helping to give them the vocabulary. You're using the word variable, independent, control, hypothesis, intensity, error, values, reliability. You're introducing these words to them in these sentences and asking them to do quite simple tasks by filling in the blanks. But what they're doing is actually, building up their vocabulary and hopefully building up their confidence. Once you've built that, then you can give them just a straight plan and investigation to do such and such. Evaluate these results from an experiment that we did. And they are important skills, especially if you hope your students are going on to A level, these sort of skills that they build here are still used and are used even more at A level. So it's important that they build those through. Okay, I hope that's been valuable to you to sort of look at those different sort of patterns. So let's just sort of pull it together and remind ourselves about the question styles we've referred to. So multiple choice, and they can be AO1, AO2, or AO3. Short answer, perhaps 1 or 2 marks, 3 marks maximum probably to define it as a short answer. And the command word for those short answers, describe, suggest. Longer answers, mini essays, which might be worth 4, 5, or even 6 marks. And they will come through with perhaps higher level command words like evaluate, discuss, plan. Experimental questions, AO3 questions, and we'll look at this idea of combs in a minute. Maths questions. And then the first question on paper 2 is always a comprehension question. And the comprehension question, is a useful skill to build for A level, where they're not necessarily a straight comprehension, like what did it say on line 23, but it could be in line 23, such and such a word was used. What's the meaning of that word? Or why do you think, in the second paragraph, they observed these certain features? Why didn't they observe something else? So again, it's very much AO2, putting things into a context and using that style. So those are the thing: multiple choice, short answer, long answer, experimental, maths, and comprehension. They're the range of questions that you can reasonably expect. From a multiple choice point of view, there's going to be more on paper 1 than paper 2 because of the comprehension taken up on paper 1, on paper 2. And also, of course, paper 2 is slightly shorter in terms of time. So a lot of people think the multiple choice is sort of like easy, but don't be fooled. Multiple choice questions can be quite demanding, and they won't necessarily just be on the recall of AO1 knowledge. You could even have a multiple choice question on maths. You could give them a question to work out, and then the answers could be picking out of 4 what the correct answer is. So, it's a very wide skill with multiple choice. And there's some different styles here. So here's a multiple choice based on a food web. Again, I think the technique here, if I do this one with you, first of all, look at the information. Don't rush to the question. So the diagram shows a food web. Yes, it does. I can see that. So what's going on? Well, I've got grass and corn at the bottom. Oh, yes, I remember my teacher told me that. What were they called again? Oh, yeah, they're the producers. They're the ones that are photosynthesizing. So the next level would be the primary consumers, and they're being fed on by rabbit or a grasshopper's feeding on both of them, and a mouse. And then the secondary consumers, I've got 2 at the top there, an owl or a snake. The owl seems to be eating either the rabbit or the mouse. The snake seems to be eating the grasshopper or the mouse. Poor old mouse is eaten by both of them. Okay, I've got the information in my head. Right, now what's the question? Which of these organisms is a secondary consumer? Well, I've made that quite easy because I've been thinking out loud and I now know what the answer is. But I'm not just going to rush to owl and pick it. I'm going to check. So A, corn, no, definitely not. Not down the bottom there. It's not even a consumer. Grasshopper, yeah, that is a consumer, but it's not on the second level. Mouse, same as grasshopper. So even if I couldn't do this question, if grasshopper and mouse are on the same level, how could one of them be right and the other one be wrong? So it can't be B or C. So perhaps I can limit my choice to just A or D. And yeah, I can go for D. So you can see what I did in terms of thinking out loud there. I absorbed the information. I looked at the question. I went for the answer that I thought was right. And then I double checked that the other three were definitely wrong. And that is a more reliable way of making sure you score well on multiple choice questions than it is simply just to dive into the question, scan over the information, dive into the question, spot the right answer, write it down, move on. Okay. We don't get a lot of kids running out of time in exams. There's usually enough time for you to do what I've just done and not feel under pressure that you're going to run out of time if you do it that way. So short answers, clearly a straight AO1 here. Describe the role of enzymes in genetic modification. 2 marks, look at the number of marks available and try and make sure if it's a description that you've said 2 things. Longer answers, a bit more context here. A balanced diet should include the correct proportions of each component. 2 of these components are vitamins and minerals. Describe the functions of the other in bold. So we'll look at this question in a bit of detail and we'll look at some samples. So just think for a minute what the mark scheme might look like for this. If you've got a pen or paper, sort of jot it down. Don't try and put it in the chat, it'll be too complicated. But just think for a minute. If you were an examiner or if you as a teacher had made up this question, what would the answers be? What are the mistakes that the students might make? So if I think out loud with you, a balanced diet should include the correct proportions of each component. Okay, that's just telling me what balance means. Right, it's giving me 2. 2 of these components are vitamins and minerals. So there's no point me writing anything about vitamins and minerals. I'm not going to get any marks for them because I've been given those. Describe the functions. So not just my question and naming them, but describe the functions of the other in bold components. So I've got to identify the other components of a balanced diet. So that's AO1 really, isn't it? What are the functions of a balanced diet? And then eliminate vitamins and minerals and talk about their functions. Okay? So I asked you to sort of think about what the mark scheme would look at. Here's the mark scheme. So a description, and remember it's for the functions. So the carbohydrates for energy or for respiration, 1 mark. The lipids, the fats for energy, but I'd also accept that's what the slashes mean, alternative answers. For storage, insulation, myelin sheath is made up of lipids. Many hormones require fat-based molecules to build them. Protection around organs. So quite a few possible answers for lipids. Protein, effectively growth and repair. Enzymes are proteins, though. Antibodies are proteins. Neurotransmitters are proteins. So they would be acceptable answers as well. Water, and that would be to transport substances. Many reactions will only happen in solution. Temperature regulation through things like sweating, prevention of constipation, helping with digestion, all possible answers for water. And fiber, roughage or peristalsis, moving food through to prevent constipation and to help with digestion. So that mark scheme is in your delegate booklet if you've been able to download it, so you can find it there. What I'm going to give now is show you two questions, which I'm going to ask, two answers, which I'm going to ask you to mark. So this was the question. A balanced diet should include the correct proportions of each component. Two of these components are vitamins and minerals. Describe the functions of the other components. I asked you to think about the mark scheme, and now I'm asking you to apply this particular mark scheme. So I'll jump back to it on a couple of occasions, but let's do A and tell me how many marks you think it is. So the mark schemes are carbohydrate function, fat function, protein function, water function, fiber function. Right, let's have a look at answer A to start with. If you could put in the chat box how many marks you would give answer A. It's a good question from Roma in the chat about why words are underlined on the mark scheme. If they're underlined, that means you don't have to use that word, but it has to be in that context. So obviously, if you just said for energy, you can't have the mark for just energy. You had to link it to respiration and carbohydrate or so on. So it's showing that it has to be within that context. Don't worry about B yet, just do A. Let me flick you back to the mark scheme again, just as a reminder, if you haven't been able to download the delegate booklet. Right, there's the question again. Someone is asking about ECF in Edexcel. If you're not sure, ECF is error carried forward. And yes, that does apply. So for instance, in a maths question, they might be asked to, I don't know, work out some sort of percentage or something. And if they had to take the percentage out of the table, and again, I'm making this up, if it was like 40%, and then they did the maths and got the right answer, that's fine. But if they'd sort of like accidentally on the first part taken the 40% and then taken it away and done 60%, and then done all the correct maths, but based on 60% rather than 40%, they would end up with the wrong answer. So they wouldn't get full marks, but they could get some marks because they've done the right maths. They just made one mistake and then did the right maths on that mistake. And that's called ECF, error carry forward. And yes, that would be true. So yeah, getting quite a range in here. And obviously I've deliberately picked one that was a little bit sort of tricky. So humans need carbohydrates, proteins, water, and fiber. Now, exactly the first step I would say to my examiners is be very careful. Is that worth any marks? No, it isn't. Because the question said, I'll go back again, describe the functions of the other components. They've just listed the other components. There's no functions there at all. So that first sentence is not worth any marks at all. They also need plenty of lipids. That's still nothing. For energy. Right, for energy. So there is marking point 2, the function of lipids. So there is 1 mark. Iron is important for hemoglobin, and vitamin A helps vision. That's true. But we were told vitamins and minerals, and we were asked for the other components. So that's no marks because they're vitamins and minerals, and we were asked for the other ones. So iron is important for hemoglobin and vitamin A to help vision. Vitamin C, again, that's not what we were asked, are also important for stopping scurvy, and vitamin D. And calcium are needed for. So some good facts in there, but they have not answered the question. Okay? So this was worth 1 mark. And some of you did give it 1 mark, but I think some of you fell into the trap. And it's difficult on screen because I took the question away from you. But if you were marking it on paper in a real exam, the question would be staring you in the face with this answer underneath. So that was a bit sneaky of me, but I hope it sort of made the point that they have to answer the question, not just write down what they know. There's no half marks in Pearson, by the way. Roma was asking that. No, there's never any half marks. I mean, obviously, if a question's worth 2 marks, they might get 1, which is half the marks, but they wouldn't get a half mark for this and a half mark for that, which would add up to 1. No, that doesn't happen. Okay, have a look at question B then. I'll give you a couple of minutes to see how many marks you'd give question B. And Gistu was asking about the balance between different questions. Yeah, any paper will have AO1, AO2, and AO3 in it on that balance 40-40-20. So you don't have a whole paper, which is all AO1 or all AO2 or all AO3. You could create that in Exam Wizard if you wanted that to help the students practice. But no, that's not the type of exam you get. Okay, I'm conscious of the time. I don't want us to run out. So yes, most people have given this 2, and I completely agree with you. So you've got starch. Well, that's not naming one of the components. That's just an example of a carbohydrate. And fats are important for energy. So yeah, fats for energy. That's marking point 2, 1 mark. Too much sugar, though, will cause obesity. That's not relevant. Proteins and amino acids are useful for growth. Yes, that's marking point 3, growth and repair. So that's 2 marks. Fresh fruit and vegetables are important for peristalsis. No, that's not a component of the gut. They prevent constipation. So correct function, but it didn't link it to carbohydrates. So 2 marks. Yes, I agree with those. That said 2 marks for this. So when it comes to experimental questions, you can use this idea of corms if you haven't seen it before. Most experimental design questions will be a full 6 marks, so it's important they can learn those. They have to sort of develop a valid experiment, and they're not just focused on the core practicals. If they're asked to design an experiment, it won't be from a core practical. So corms is one way of doing it. It's not the only way of doing it, but it gives them that sort of prompt. It gives them the idea of what is it that you're going to have to change? What is it that you're going to have to control? Because this is biology, what are the organisms? What is the biotic factor? You know, are they all the same species? Are they all the same type of size or age that comes into it? How to repeat Have you only got one reading, in which case it could be some sort of one-off? What is it that you're measuring? And sometimes there'll be more than one thing. So, there might be 2 marks available here. And then what do you need to keep the same? What's the controlled variables that you need to throughout the experiment? So you can use this system in order to design a practical. So be clear about being set up for the C mark. State factors, not just the same animal, state that it's the same species of animal or something. Don't use vague terms like size or amount. Use mass, length, volume. The reliability, the repeatability of what you're doing. You're not adding additional values in. You're not doing it, if you're repeating it, you're not doing one at one temperature and one at a different temperature. That's something else. You're repeating it at that same value to check you get the same results. What to measure? As I said, there's often more than one things to measure, so there could be more than one mark there. And equally, what you have to keep the same. There could be more than one mark there. So here's a typical question on this. Plant growth substances stimulate root growth from a cut stem. Describe an investigation to find the best concentration of plant growth substance to stimulate root growth. You should include experimental details in your answer. So have a think for a minute what you think a mark scheme would look like for that. And here's a mark scheme using the idea of columns. So obviously you'd have to use different concentrations of auxin. You'd have to make sure the plants were the same species, or even cuttings from the same plant or on the same plant. You need to repeat it. What would you count? Well, you're working out the effect that it has. So you could count the number of roots, the length of the roots. You measure it with a ruler. You'd have to leave it for a certain amount of time. So is that being cleared? And then again, what would you need to keep the same? Well, the temperature, the oxygen level, the light level, growing in the same media, the same humidity, same minerals available to it. So again, it's a bit tricky not being able to keep the mark scheme on the screen for you. But have a look at this question and see what you think. Perhaps even if you don't come up with a mark, which of the columns do you think they've hit when you're doing this question? You can put it into chat if you want, although, as I say, if you're finding it a bit awkward, then don't worry. So would you give this the C mark, the change mark? Would you give it the O mark, keeping things the same? Would you give it the R mark for repeat? Would you give it the M mark for what they're measuring? Would you give it the S mark? So if we go through it, I would take several oat seedlings. Okay. So that could just be the O mark because they're saying oat seedlings are being quite specific. I will grow them so that their roots begin to develop. Nothing there yet. I will then add a range of different auxin concentrations. So there's the C mark on each of these roots. I will repeat each concentration with 3 plants. So there's the repeat mark. So that's 3 marks so far. I will put the plants into soil, just soil, there's nothing there, to see how much they grow over a constant. No, you've got to say a day, be specific. So it's not a mark, a measure mark there. I will keep everything the same. No, sorry, not good enough. What are you going to keep the same? Not just keeping everything the same, such as the amount of nutrients, too vague a term, nutrients. So I'm going to give it 3 marks for the C, the O, and the R. Right, what about the next one? Sorry, take it back. That was a bit too fast. Yeah, we're getting some high marks coming through here, which is what you'd expect. So if I take you through this one, I will make a range of concentrations of auxin. So there's the C mark. I will then take plants of the same species, so there's the O mark, and same age, so even better, and place the different concentrations of auxin on the roots of each one. I will repeat this. I'm not just saying they're repeating it, they're saying I will repeat it 2 more times. So yes, they can have the R mark, so that there are 3 of each concentration. I will measure the length of the roots. So there's an M mark for all of the plants. I will put the plants into the same soil, that wouldn't be enough, with the same compost, brackets, or even better, same mineral ions concentration. Yes, there's an S mark. I will measure the lengths of the roots 1 week later. So a time, so there's another M mark to see how much they have grown. I will keep the oxygen and carbon dioxide concentrations the same. Didn't say how, but yeah, that would probably get them another S mark. So yes, I agree with nearly all of you there. Definitely a safe 6. Right, let's look at one more. Yeah, majority of you are coming up in the 2-3 region, which I'd agree. It's certainly nothing like as good as the previous one, is it? So if I go through with you, take 2 plants of the same species. Yes, that can give me the O mark. Place the roots of one in plant hormones. That's not good enough yet, but not the other. No, it's the mark scheme, if you remember, I said not just all or nothing. It had to be a range. So nothing there. The plant hormone should make the roots grow longer. Well, that's not telling me anything practical than the one without the hormones. Again, true, but it's not telling me anything. This is because the plant hormones affect the speed at which roots and shoots grow. Yes, wandering off the question, not good enough. The hormones used could include growth auxins, which also affects phototrism. But I could word spot the word auxin and think that's worth them are, but they haven't actually said what they're doing with it. I will repeat the experiment. Okay, we can just give them the repeat mark there, even though they've chucked it in at the end. So probably 2 marks. That's what I'd agree on that one. Okay, so practicals, I hope that's given you some idea there. Graph questions, there's usually at least one graph question on the paper somewhere. Here's a list, you can use the the term slap or slap you or slap UK or whatever if you want to. But basically, here's a checklist which is useful for the students to sort of say, if you've drawn a graph, you know, have you obeyed these rules? Is the scale linear, half on each axis? Are the lines straight between the points and neat, or have you been, have you been asked to draw a bar chart? If you've been asked to draw a graph, then there wouldn't be bars. Are the axes the correct way around? Have you labeled the axes? Has your label on the axes included the units? Have you plotted the points carefully? Normally you get a plus or minus one small square tolerance. So have you plotted them accurately? As I say, if you've got the units on the axes, and if there's more than two lines, or one, if there are two lines or more, then you've made it clear which one is which by labeling them. So some basic rules there on graph questions. Calculations, about 10% of the marks will be maths questions based on calculations. Percentage increase, they often mess up on, being not quite sure which number, getting what the rise is or what the change is, and then which number they put that over in order to get the percentage change. Here's another one with a set of data, and you're asked to plot the graph. And it's telling you here, join the points with straight lines. And here's one where they've got no data at 50. So if we look at the mark scheme for this one, if you think what the mark scheme would look like, see it's worth 5 marks. Just have a little think what you think the mark scheme would look like. So there you've got it sort of set out with that slap idea, although it doesn't have to be set out like that. So have they got the marks for the linear scale? Have they drawn the lines correctly? Are the axes correct? Are the axes labeled? And have they plotted correctly? So you could share that with your students and let them see how a graph would be marked. And then here's a good example. Clearly these students have got that spot on. They're able to sort of, the axes are sensible, 20, 30, 40, 50. Doesn't have to start at 0. Axes up the side, labeled, minutes, equal units going up. I won't go back to the information, but the plots are accurately plotted and the lines are accurately joined up. So this would be worth the full 5 marks. Be very careful about tram lines. If they make a mistake when they're drawing a line in, rub it out carefully and clearly and put the next one in, or clearly cross it out. Don't just leave like 2 lines, because the examiner will penalize you for tram lines. Comprehension questions. As we said, if you look at the question on paper 2, the first question will be I think I've got a sample of that, but it wasn't in your documents. But you can go to any paper 2 and just look at the first question. Go to exam questions, exam wizard, look at the first question on any paper 2, and you'll see the idea of the comprehension. And it's often synoptic. In other words, it's often dipping into, it gives a way of dipping into lots of different parts of the specification. All questions will have a command word, and this is common with what we did on module 1 now. So the command words are the instruction of what to do for that particular question. And you'll find all the command words are in the specification. And it's a good idea to perhaps share that with the students. Here's a table that's actually in the specification. I think it's appendix, is it appendix 5 or something at the back? It's certainly one of the appendices at the back. Yeah, it is. It's appendix 5. And so you can share this with students and help them to understand that both the specification and the exam papers will have command words. It's important that they understand what they are, and it's important that they understand they have levels of difficulty. So an explain question is more difficult than a describe question. A state, a name is quite a basic one. When it comes to evaluate and discuss, it becomes more difficult. So here's a sort of sample with a describe and explain, where you've got the first part with the table, and then explain the differences coming up. Remember, I said to you, the key thing in explain is to be able to actually get the word because in there, because to explain something, you have to say this happens because. You might get a command word which is comment on, which is obviously going to be much wider in terms of what it looks like. So here's a load of data. And as I said to you earlier, we don't really have time to do it now, but you'd have to go through that data, get it into your head. And then, as I say, the question is just asking you, the scientist concludes that the hot water pollution affects the population of fish. Comment on this conclusion. If you're commenting on it, it doesn't necessarily have to be true or false. It may be inaccurate. And you can see the mark scheme is listing possible comments that could be made on that. Suggest questions also are open. So suggest questions could be to use your knowledge and think about solutions to the problem as presented. And don't think students, when it comes to suggest, are going to think there is one right answer which they have to get right. There won't be if it's a suggest. It literally is asking their opinions based on the information that's being given. Evaluate equally takes you through things. So here's a question, which you may have used with your students. The actual here is a sort of the bell jar example of showing how the lungs inflate due to the pressure inside the thorax and the action of the diaphragm moving up and down. So this is a standard model that was used. And there's a question on this one where you're asked to evaluate how useful that is. So, how useful are the balloons as an example of lungs? How useful is the bell jar as an example of the rib cage? How useful is the rubber sheet as an example as the diaphragm? Tube A and tube B as a trachea and bronchus. So you can see the mark scheme would sort of give you 4 possibilities there. And it's wide open because it's an evaluate. So it's looking for some sort of reference to the diaphragm as opposed to the rubber sheet, to the balloons, as I said. Discuss command words. Again, much more open. So you've got a question here where there's some data presented and the student's drawn a conclusion based on that data. And then they have to sort of come in. There's a mark scheme on that. So you do have a copy of these slides. I'm conscious I'm starting to rush a bit because we're going to run out of time. But you do have a PDF of all these slides. So you can go back later and have a look if you want afterwards, if you feel I'm rushing through this a little bit too fast. My apologies. So again, when it comes to actually sort of marking scripts, I think I might skip over this one because it's quite a complicated one on eutrophication. So you've got a sample question here and a couple of sample answers. And this is about sort of genetic modification. This is a good answer on this question. If you want, I don't think we've got time to go through it in detail, but it did score all marks. And this is a less good answer. This one was really, you could argue whether it would only get 1 mark. You might possibly give it 2 marks. This is the one we were looking at. And here's a sample question on that. So you can see, the question was about ventilation and the students going on about gas exchange here. So really the only thing they're going to, the score there is in the last sentence. There are no chest muscles on it, fair enough. The balloons are like the lungs and the glands are like bronchioles. So, you possibly just get the 1 mark there for the balloons are like the lungs. So not such a good answer there. You look at this answer. You can see this would pick up all 4 marks because they've got the second marking point there. The balloons are like the lungs. The trachea splits into the bronchi. The sheet at the bottom is like the diaphragm. And they've grouped it into what they think are accurate sort of interpretations. And then the things that are not so good, that, the bell jar doesn't move like the rib cage would. It's not broken up into separate ribs. There's no intercostal muscles. The lungs don't fill the whole space of the jar. And then they've made an overall conclusion that they think it's a good model. So there's a really, two of them. And you can, take these away and use them with your students. Do the same exercise with your students that I've done with you to sort of look at what's going on there and to make those comparisons. Okay? So you could give the students, show them that model. If you've got that as a bit of equipment, it's not quite easy to make up. So you show them that model. You tell them what the various parts represent. You then ask them to think, well, if you've got a question about this, to evaluate what's going on, what are the various bits that you think you could write about? What about the tube going down through at the top? About it branching? About the balloons? About the bell jar? About the diaphragm? Having asked them to sort of think about a mark scheme, show them this mark scheme and show them that it's a 4-mark question, although there's actually more than 4 marks available. So you could get full marks even if you don't cover everything. Then ask them to mark an example. It's anonymous, so they don't know who it is. It's not somebody in the class that's going to be embarrassed. And discuss with them whether it's a good answer or not. And then show them this one, which is clearly a much better answer. So you're sort of helping to reinforce what that was all about. You're teaching about the lungs, but you're also helping to sort of develop their own exam technique. So I hope they would all be ways that you could use with students in terms of developing those techniques. Okay, so let's conclude now then with the overall sort of support that's available from Pearson's. So there's a table there showing you exactly what's available where. So obviously courses like this, getting started, training events, you have subject advisors from Pearson's and often in different countries. I was lucky enough to be out in Chennai in India and Colombo in Sri Lanka doing some training. And on both occasions, I met the local Pearson reps when they were there, and they were offering support and so on as we're going through. The schemes of work are published, and they were covered in Module 1. Even if you don't use them directly, they contain a huge amount of very valuable resources and links to various internet sites that you can use. Skills mapping, we've talked about this morning in terms of transferable skills and where they are. Huge amount of assessment material. You've got the original sample assessment materials when it came out, but now lots and lots of past papers, given that there's sort of 2 or 3 exams a year, and this has been going since 2018. You've got a huge bank of past papers and the examiner reports that go with those. So you could pick questions that were answered well or questions that were answered less well. And so you can access all of these, through Exam Wizard. And you can also check, as we said earlier, with Results Plus and access to scripts. So hopefully you'd agree, there's a lot of support there. The best place to go to really is this page. So you go into Pearson qualifications, you're selecting International GCSEs, you select biology, and that'll give you the sort of drill down of what's going on there. And you can see in the course materials, when this slide was made, there's still obviously only 4 in the specifications, but the exam materials have now gone up to 136, because obviously the 24 papers are in there now, and the teaching and learning materials, there's a couple of extra bits being added in there, so they've gone up to 47. So you can just open up each one of those and then click the various boxes as to what you want and download directly from it. So you can find things like in the teaching and learning materials, you'll find recordings of these courses. So, you could actually sit down if you wanted to, and you don't have to, if you don't want to, of course, and you could go through the whole course again and just listen to the recording, or you could share it with your colleagues back in schools. So that's in that Pearson qualification, that's where you'll find all those various materials. There's also various booklets published. So there's a thing called Getting Started Guide, which takes you through a lot of what the early part of this was. A very, very valuable book on the top right and in the middle at the top there. And that is the Core Practical Guide. And the Core Practical Guide is something which is for biology and chemistry and physics. Very big document. And it literally, as it says on the cover there, it takes each core practical in turn. It makes links as to what spec points are covered by that core practical. It gives you some ideas about the skills that you're developing with it. It shows you some exam questions that have been set on that particular core practical in the past. So it takes you through that very carefully. And then the other document there, the top right, the guidance on practical terminology, that's if you're unsure about words like repeat, precision, reproducible, reliability, anomalies, lines of best fit, all of those are covered. It goes through all the various terminology, significant figures, numbers of decimal places, lines of best fit. All of those are covered in that particular document. So that can be a very useful document as well. And then the Maths for Scientists, again, that's a common document across biology, chemistry, and physics. And that goes through each of the math skills. It looks at things, like probability. It looks at rates of change, looking at graphs, calculating gradients, equations on straight line graphs. All of those are covered step by step with detailed examples of helping you to teach those skills, as well as giving you exam questions that are used there as well. Okay, so very useful documents there. And then finally, bottom left, you can see there an example, cell structure and stem cells. There's 3 of those booklets. That one that's illustrated cell structure and stem cells. There's another one on GM, on genetic modification, and there's another one on protein synthesis. So if you wanted a sort of detailed subject background, those are all available. Results Plus we talked about, and again on your PDF of these slides, you can see a hyperlink on the bottom there. So if you just search Pearson Results Plus, you get there, and there's lots of YouTube videos within that take you through exactly how to use it. You can load mock results into it and use the facilities within it for that, but it is primarily used, as I say, when you've had one cohort of students go through. You can analyze how your students compare perhaps to other classes in the same school or other schools in your area. You can't name the schools, but you can do comparative of your results to others. And of course, you can compare them nationally. So it gives you quite a sophisticated tool as to what you can do in analyzing the results that have already happened for students that have gone through. Not a lot of use to you now, those of you that are new, we haven't got any students that have done it yet, but once you're in that position, then you can go through and look at all of that. Exam Wizard we talked about, so I won't go through that again now. Access to scripts. This gives you a chance to, as I said to you earlier, go straight in and have a look at a student. So a student comes back to you on results day and said, oh, I got a grade 5. I'm really disappointed. I hope they get a grade 6. Or you can go in, look at their actual paper, see what marks they got. You can look up to see how close they are to a particular boundary and decide if you want it. Additional resources like, the school textbooks. And if you buy the textbooks, it automatically takes you in to a link to the active online version of the textbooks. You get a code inside the book that allows you to sort of access that. And then teaching hubs, there's a picture of the textbook. And teaching hubs are available. And this is something, a new development from Pearson's, which allows you to go in and access various materials from there. So my apologies for rushing a little bit, but I know we get sort of cut off at the end. So there's Irena, who is there to sort of be able to help you where she needs it.